Today, we're diving into one of the most ancient, most fundamental processes in all of biology. It's the story of how our bodies turn food into fuel. We're going to unpack the incredible biochemical factory known as glycolysis.
You know, it really all boils down to energy. From a muscle contracting to a single neuron firing off a thought, every single thing your body does requires fuel. But how do you get from, say, a piece of pizza to a brilliant idea? Well, that journey starts with a single heroic molecule.
So, our story kicks off with something we all do every single day: eating. The carbohydrates in our food, bread, pasta, fruits, are the main source of our body's absolute favorite fuel, a simple sugar called glucose. But first, we've got to break our food down to even get to it.
And this journey starts the second you take a bite. Seriously. Enzymes in your saliva, like amylase, get to work immediately, just chopping up those long, complex starch chains. Then, down in the small intestine, pancreatic enzymes jump in to finish the job, breaking everything down into simple sugars. The most important one for us is glucose, which gets absorbed right into your bloodstream, ready to be shipped out to trillions of hungry cells.
Okay, so our glucose molecule has hitched a ride through the bloodstream and has finally arrived at its destination, a cell. But it can't just waltz right in. It's got to get past the bouncer at the door, the cell membrane. To get inside, glucose needs a special pass. It relies on these protein channels called glucose transporters, or just GLUTs for short. Now, you can think of these like highly specialized doorways, each one designed to let only glucose through. But here's the thing, not all doors are created equal.
And this is where it gets really interesting, because different cells have totally different needs. Your brain, for example, uses something called GLUT3 transporters. These things are like a powerful vacuum cleaner. They are incredibly good at sucking up glucose from the blood, even when levels are really low. It guarantees your brain gets a constant, steady stream of fuel. But then you have muscle cells. They use GLUT4, and this doorway is locked. It only opens up when the hormone insulin shows up with the key, basically telling the muscle, hey, we just ate, time to grab some glucose.
So, once our glucose hero makes it through the door, the cell immediately plays this super clever trick on it. An enzyme comes along and slaps a phosphate group onto the glucose molecule. This whole process is called phosphorylation, and it does two crucial things. First, it gets the glucose all revved up for the next steps, but more importantly, it traps it. The cell's doorways don't recognize this new version of the molecule, so the glucose is now locked inside, completely committed to its new fate.
All right, so our glucose is trapped inside the cell. Now the main event can finally kick off. This is the priming phase, where the cell has to get the glucose molecule ready to be split.
And here is the first big twist in our story. It sounds a little backward, right? But to make a whole lot of energy, the cell first has to spend a little. You know the saying, you've got to spend money to make money? Well, it's the exact same principle.
So what's the price of admission? Two molecules of the cell's energy currency, ATP. The first ATP molecule was used to trap glucose inside. A second one is used now to add even more energy, making the glucose molecule unstable and getting it ready to be split right in half. Okay, the initial investment is paid. Our 6-carbon glucose molecule is perfectly primed, it's unstable, and boom, it splits into two 3-carbon molecules. And this right here is where the cell starts to see a serious return on its investment. The energy harvest is officially on.
The whole second half of glycolysis is all about the payoff. These two smaller molecules get put through a whole series of reactions that are designed to wring out as much energy as possible. First up, the direct cash payout. In a flurry of rapid-fire reactions, the cell generates a grand total of 4 ATP molecules. Remember, our one glucose molecule split into 2, and each of those halves gives us 2 ATP, so that's a total of 4. But that is not all. The cell also captures these high-energy electrons in a special carrier molecule called NADH. You can think of NADH as a little rechargeable battery. It doesn't actually spend its energy right here and now, but it carries it off to another part of the cell, the mitochondria, for an even bigger energy payout down the line. We get 2 of these charged-up NADH batteries.
So let's do the quick math. The cell invested 2 ATP, but it generated 4. That means the net profit from just one molecule of glucose is a modest but absolutely vital 2 molecules of ATP, plus those 2 high-energy NADH batteries that are saved for later.
Now, the end product of this whole 10-step factory line isn't just energy. Our original glucose molecule has been chemically rebuilt into two molecules of a compound called pyruvate. And pyruvate now finds itself at a major fork in the road.
What happens next depends entirely on one thing. Is oxygen available? If oxygen is around, the cell goes the aerobic route. Pyruvate gets sent into the mitochondria, the cell's powerhouse, where it can be completely broken down to generate a massive amount of extra ATP. It's the slow and steady, but incredibly efficient path. But if oxygen is scarce, like when you're in an all-out sprint, the cell switches gears to the anaerobic path. Pyruvate stays out in the cytoplasm and is quickly converted to lactate. This is way less efficient, but it's lightning fast, giving you those quick bursts of energy you need for intense effort.
For the longest time, people thought lactate was just a metabolic waste product. You know, the stuff that makes your muscles burn. But we now know that is not the whole story, not even close. As this quote says, lactate is actually a super valuable fuel. It can be shuttled out of the muscle cells into the bloodstream where other tissues like your heart and even your brain can grab it and use it for energy. The liver can even recycle it back into glucose. It's a huge part of our body's incredible energy economy.
But here's the thing. This entire assembly line doesn't just run on autopilot. No way. It's exquisitely controlled. The cell has to be able to speed it up or slow it way down depending on its immediate energy needs.
So that brings up a really important question. How does the cell act like a factory manager, controlling the production line to perfectly match supply with demand?
The beauty of the system is all in its feedback loops. There's this one key enzyme, PFK1, that acts like the main gas pedal. When the cell is flush with energy, ATP itself acts as a brake, binding to PFK1 and telling it to slow down. But when energy gets low, a different molecule called AMP builds up and slams on the accelerator. On top of that, hormones like insulin can send system-wide signals to ramp the whole process up. It is a beautifully tuned system that makes sure the cell produces exactly what it needs, precisely when it needs it.
Now, understanding this process isn't just some abstract science lesson. It has massive implications for our health. For instance, many types of cancer cells are basically metabolic monsters. They completely rewire their systems to rely almost exclusively on this fast anaerobic version of glycolysis, even when there's plenty of oxygen around. This is called the Warburg effect. They crank the pathway into overdrive to churn out the energy and building blocks they need for their rapid, uncontrolled growth.
When you really think about it, glycolysis is ancient. It's a biochemical fossil that connects us to the earliest forms of life on this planet. It probably evolved on an early Earth where oxygen was almost non-existent, which is why it's the universal foundation of energy metabolism for nearly everything alive. So, we'll leave you with this thought. When a process so central, so fundamental to life itself is disrupted or hijacked, the consequences can reshape our very biology.